All right, so we're going to have a fun conversation on our Science and You segment that we do. As always, joining me is my co-host, our chief science advisor for our program, physicist Dr. Yu. How you doing?
I'm doing great. Thank you for having me on. Yeah, it's
And
a
I
nice.
and
Yeah,
it's great to see you as always. But we are really excited for our guest today, who is a professor at biology at Tufts University, Dr. Michael Levin. How you doing?
Good. Hi, nice to meet you.
Great. Now you've been getting a nice algorithm boost. I don't know who you talk to at YouTube, but you've been getting lots of suggested videos. Every time I'm on YouTube, it's Lex Friedman, it's all these different programs interviewing you, and you've got some interesting ideas. So I'm looking forward to talking to you.
Thanks so much. Yeah, happy to talk to you. I know nothing about YouTube or the
Yeah,
algorithm.
I know I'm teasing you. I'm being facetious, so I'm trying to break the ice there. But anyways, I think it's a fascinating topic, the topic of bioelectricity, which we wanted to have you on. It's something that Dr. You and I have explored in different fields and different guests, and I'm looking forward to learning your insights into this. I know that there's folks out there who I've talked to, like Dr. Rupert Sheldrake, who's proposed morphic fields, and I know that he gets derided in a lot of circles, but I don't really care. I'm not trying to play politics and academia. That's not my world. I'm here to learn the truth and explore it. So I'm grateful for the opportunity to hear from pioneering scientists such as yourself. So for those who are not familiar, I want to dive into some of the implications of your work, but in general, what has been the distinctive focus of your research at your university?
Well, my group fundamentally works on different kinds of intelligence in various substrates. So we look at memory learning, decision making, problem solving in all kinds of unfamiliar substrates. So cells, tissues, organs, slime molds, molecular networks, various kinds of biobots that we make, all these different things. And one kind of a very important phenomenon is that of cognitive glue, which are basically just a set of policies and mechanisms that allow parts to work together towards some kind of a large scale whole that does things that the individual parts don't do. So in our brain, the reason that you and I are not a pile of neurons, but something more than that, is that there is this cognitive glue mechanism, aka electrophysiology, which binds the neurons together into a coherent being that has plans, memories, preferences, goals, and so on that no individual cell has. So bioelectricity actually does this very widely across life. It was here long before brains and muscles and neurons and those kinds of things appeared. And so that's one reason that we study bioelectricity, is to understand how it is that cells come together to form complex organs, to repair them, to suppress cancer, to form embryos, to regenerate. And once we understand how that works, we can then communicate with that collective intelligence of cells, impose novel goals that will be useful for healing, for repairing birth defects, for normalizing cancer and things like that.
Yeah, I've seen some of your talks about the implications for cancer. Are you doing research in cancer yourself, or just proposing, theoretical work, or where does that stand for you?
Oh no, absolutely. We do. Yes, of course, we do research in cancer. We've shown over the last, 15 to 20 years, we've shown that you can, in animal models, you can detect regions where tumors are going to form. Okay, early before they become histologically apparent. We've shown that you can induce, so for example, something that looks very much like metastatic melanoma in an organism without any kind of carcinogens or mutations or DNA damage or any of that, simply by disrupting the specific electrical communication between cells. And most importantly, we've shown in these animal models that if you do put in nasty human oncogenes, things like KRAS mutations and so on, you can actually suppress tumor formation by managing the bioelectrics of the cells correctly. Doesn't kill the cells, doesn't fix the mutation, but it basically it's the physiology that drives. It's not the genetics that drives. And so despite some of these genetic defects, you can still get perfectly normal cells out of it. So yes, this is we do the vast majority of my lab does experimental work.
Right. Now, you've also mentioned this Platonic space concept. This is something that we hear about in philosophy, but this is something that you think is very real. What is that? Is that a synonym for the cognitive glue or something else?
No, it's not the cognitive glue, it's something else. Typically, when you look at any kind of a living organism, a standard natural living organism, and we ask why does it have the shape that it has, why does it have certain behaviors? So in other words, patterns of form and function, right? Different patterns, anatomical and behavioral patterns. And you ask why does it have these specific patterns? The answer is usually, well, evolutionary history. In other words, eons of selection for a specific form and function. The reason I've started speaking about patterns from the Platonic space recently is that we can now make novel synthetic beings. And so, of course, people have made chimeras and various biobots before. We make something called xenobots, which are made out of frog cells. We make anthrobots that are made out of adult human cells. And all of these novel creatures, they have new forms, they have new behaviors, they have new capabilities. We can talk about that if you want. But the answer to where did those come from is not evolutionary selection, because there have never been any xenobots. There have never been any anthrobots. Nobody got selected to be a good anthrobot or a good xenobot. So the question becomes, if we can't infer the possible forms and functions of beings from their evolutionary history, where do they come from, right? And the question of where do patterns come from is pretty deep, and it's been studied for thousands of years. And so Pythagoras and Plato had already seen that there are these mathematical patterns, facts of mathematics, facts of number theory, facts of geometry, things like that, that matter in the physical world. They guide things that happen in the physical world. Biology exploits them all the time, and we could talk about some examples of how that works. But these are the thing about these patterns is that they are not determined by any facts of the physical world. In other words, there are specific facts about numbers, about the way that mathematical objects relate to each other that would not change even if you changed all the different constants at the beginning of the Big Bang, right? So this is a view that many, not all, but many mathematicians kind of adopt, is this Platonic view where these are facts that are independent from the reality and from the parameters of the physical world. They do not derive from the structure of the physical world, nor can you change them by anything you do in the physical world. And so the space of those kind of patterns, the mathematicians call it a Platonic space. My more recent attempts to develop a formalism for understanding where the form and function of new beings comes from basically links to those ideas and suggests, and this is very, of course, this is very speculative and so on, but what I suggest is that Platonic space has not only the facts of mathematics and geometry and computation and things like that, but it also has much more complex active forms that we would recognize as kinds of minds and kinds of anatomical forms. And so what we're doing, the question in biology is as to where does this specific set of shapes and behaviors comes from? Well, partly genetics, partly environment, but partly something else, which is neither of those things, and that is the laws of mathematics. So there are inputs into biology that come from wherever it is that the laws of mathematics come from, and the standard name for that is the Platonic space.
And would you say that mathematics is that consciousness proceeds from mathematics, or mathematics is an extension of consciousness, or is that, we're not determining that yet?
Well, yeah, I'm not exactly sure how to answer that question. I mean, I do think that if we're going to add, and okay, so I don't work on consciousness, all right, and I don't talk about consciousness much yet. But if I had to say something about this, I would say that basically when we have patterns ingressing into the physical world that we recognize as kinds of minds, that is behavioral patterns of behavior, patterns of information processing, behavioral propensities. When these things ingress into the physical world, consciousness is what it feels like from the other side. In other words, standard third person science is what we see from this side. So in the physical world, we can observe these things happening, but minds are actually are patterns that inhabit this Platonic space, and consciousness is what it feels like to be one of those complex patterns when it is interacting in the physical world. That's about as much as I can say about it right now.
Yeah. Dr. Yu, do you have any questions?
Yeah, the professor said something very you combined so many deeper concept. Can we can I ask some basic questions? You mentioned about what is called cognitive fluids. Is that right?
Glue, like sticking together, glue.
Cognitive glue. Okay. So I watched some of the video you talking about, some kind of universe. There's some purpose, right, from the cell or from the integration of something. Can you explain what is the bioelectricity?
Yeah. So
You believe that's the energy how we function? Is that right? Yeah.
well, specifically, what I'm saying is that we know that in the brain, the reason that you and I have thoughts and goals and memories and preferences that our individual neurons don't have is because there is a process that binds all of those neurons together to do computations and other things in other spaces to which neurons don't have access to. So, for example, you may have social goals, you may have financial goals, you may have all kinds of strategies, planning, memories, and so on in spaces that your individual neurons don't have access to. And the reason that works is because these neurons are combined into a network that processes information in a particular way. The thing that allows the neurons to be part of that network is what they call electrophysiology. It's what neuroscientists call electrophysiology. It's the ability to construct an electrical network that can do some very interesting things. In particular, it can be the substrate for some really interesting patterns of computation and behavior that we call a human or some other kind of personality. Well, if you ask where did that amazing capability come from evolutionarily, it turns out that the way that neurons do this with ion channels, which allow every neuron to have a certain voltage parameter across the membrane, and electrical synapses, which allow them to exchange voltage across the network, so the voltage can propagate through that network. They have a special electrogenic machinery, meaning they have ion channel proteins on their surface, which allows them to have a certain voltage state. They have electrical synapses known as gap junctions, which allow that voltage to propagate through the network under specific rules. And if we ask where that amazing machinery came from, we find that through evolution, it's actually extremely ancient. So it goes all the way back to unicellular microbes. So bacteria and bacterial biofilms were already using bioelectricity, so the ability to form electrical networks that have memories to make decisions and so on. Already bacterial biofilms were doing this. So what I'm interested in is the role of these electrical networks in merging individual subunits like cells into larger scale collectives. And so now we have now seen how that actually works and how the bioelectrics allows cells to share memories. And when they share enough memories in a particular configuration, you end up with a larger scale computational system which can do things that no individual cell can do. So that's why I call bioelectricity a kind of cognitive glue. It is not the only kind. There are other kinds of cognitive glue. There is stress sharing. There are certain biochemical signals. There's other ways to do it. But bioelectricity is a very convenient way of doing it, which is why evolution uses it for neuroscience, for neural function. It's why we use it for our computational technologies. It's just a very convenient modality.
You mentioned stress sharing, and I heard you mention you could maybe talk that to our audience about how cells can share stress. And
Sure.
From an intuitive standpoint, you think about that in human beings. If someone walks in very stressed out into a room, they can share contagiously that stress. There's some various forms of communication, not just expressing a stressful attitude or whatever in words or something. Just the vibe, people call that the vibe shift or something's off. This person seems irritated or stressed. So I'm just thinking of that from the social standpoint, but from the cellular standpoint, what do you think is this shared stress, and what's the mechanism that allows that to happen? Is that an electrical mechanism?
Well, stress sharing doesn't have to be electrical. As far as we know, it probably is not electrical, but it's a very important mechanism for binding cells together towards common purpose. And what I mean by that is the key thing that underlies most of what's interesting about biology is goal directedness. It's the ability for cells and even subcellular components to do a kind of loop where it's basically a homeostatic loop. It's like your thermostat. There's a set point. You measure the state. If the state is different, then you take actions to get that error as small as you can, right? And when you reach the set point, you can stop working until it drifts off again. So that ability is really important. And so what happens? The thing that drives that loop is the mechanisms where the further you are away from your goal or away from your set point, the higher the stress level is. So stress is a way to integrate and respond to the fact that you're away from your desired set point. Let's say it's for a cell, that might be for pH or hunger level, or there's a million different things that could be off in the cell. So what you can imagine as a simple example, imagine that there's a field of cells here, and there's a cell down at the bottom, and what it's trying to do is get up to the top. And the cells typically know their positional information because there are cues in their environment that tell them where they are. And so let's say there's cells in the bottom that needs to get up here. Now, all of the cells in between there, if they are in their correct positions, they're not going to move or they're not going to do anything to let the cell get up there, because for them, their stress is very low. They are exactly where they're supposed to be. And it's very hard to get them to do anything else because they're in their correct positions. But now one thing you can do is as that one cell that's in the wrong position, one thing it can do is it can start leaking its stress molecule. And what I mean by stress molecules are all the way back from the beginning of cellular life. There have been specific molecules that are the mediators of the stress, of the fact that something is wrong. We've gotten away from that happy set point where we're supposed to be. So now, if that cell just becomes a little bit leaky to that to its stress molecule, you can imagine what's going to happen. It's going to leak out. It's going to affect the neighbors, and because the neighbors are using exactly the same molecule to signal to detect their stress, as far as they're concerned, now they're stressed out. And so So by sharing your stress, my problems become your problems. What that means is that these other cells, as they get stressed out, they get more plastic. They're more willing to do new things. And as they get more plastic and start moving around to see what they can do to reduce their stress, now the cell can get up to where it's going. This is just one simple example. There are a million other ways this can work. But the idea is that by sharing the stress, it provides, again, it's a notion of glue, because what it does is it binds us all to common purpose. Before the stress sharing, those other cells didn't care that you wanted to get up there. They were not sharing your goals. Once you start leaking that stress, now everybody's interested in the exact same goal, to get you up there so that everybody's stress can come down. So it's a way to bind multiple subunits towards common purpose in a way that doesn't require altruism or any of these kinds of mechanisms. It's just a very simple mechanism that evolution can exploit to make sure that multiple subunits are all aligned towards the same goal.
Right.
Okay, so I think I'm getting a little bit, okay. I try to learn here. So you mentioned about when people has a stress state, so the cell gonna have at a different position. You said that the cell has to move up. So are you talking about the stress caused by the motion of the cell?
No, I'm and I haven't said anything about people, so I don't, I'm not talking about people here. I'm talking about cells. Imagine the scenarios that we're interested in are where cells build a whole embryo, or where they regrow a limb. For example, if a salamander loses the limb, right, the cells will rebuild a limb and then they stop. All of those kinds of cases of remodeling, of regeneration, of embryogenesis, metamorphosis, all of those kind of processes require many, many cells to work together to build something. When you look at an embryo, why do you call it one embryo instead of 100,000 cells at an early stage? There's 100,000 cells. Why do we call this one embryo? What is there one of? Well, what there's one of is the fact that all of the cells, under normal circumstances, all of the cells are bound together by the same plan and journey that they're going to take in anatomical space. They're going to go from being a single cell, the fertilized egg, to being a shape of a human or a giraffe or a whale or whatever they're going to be. And the reason that they're an embryo and not just a pile of cells, it's the same reason that we are us and not a pile of neurons, is they all share the same goal. Sharing goals is what makes for the boundary of a collective. It's when they all share the same goal. And you need some kind of process to enable these active subunits like cells to share the same goal. It could be the memory sharing through electrical networks. It could be stress sharing through diffusible signals. There are probably many other ways to do it. But you need them all to be aligned towards the same goal. And so what I'm saying is the reason that cell at the bottom there in my example, and there's many other examples that we could say, the reason that that cell is under stress is because it's in the wrong position. So stress is what enables all of the cells to eventually land in the correct configuration to be a proper body of a proper structure. Cells that find themselves in the wrong position they know they're in the wrong position, and that's stressful to them. And morphogenesis is a kind of stress reduction scheme. So, for example, if you take a salamander and you take the tail and you graft it onto the side, the flank of a salamander, over time, what happens to that tail is that it remodels into a limb. It becomes a limb, which is right. And so the individual cells, now imagine that tail, right? So here's the tip of the tail. The cells sitting at that tip of the tail, they're locally fine. They're tail tip cells sitting in a tail. Why should they change? But they become fingers over time. And they change because all of the cells there's a large scale propagating signal that says to them, you are in the position where you are, you should be a different structure. And that's why they act. They don't just sit there, they spend energy. Many of them will actually die, many of them will divide, but they'll do all these amazing things in order to reduce the error. And the error, if you're sitting in the flank of a salamander, you shouldn't be a tail, you should be a limb. And so the ability to store a set point, a large scale set point, like we're not just a bunch of cells, we are a tail, or we are a forelimb or a hind limb, whatever we are. The ability to store that set point is what bioelectric networks allow. They allow you to have a memory of what you're supposed to be. And they also allow the cells to work together to implement that particular goal.
Okay. So I think I'm get understand a little bit more. I'm a physicist background, and I'm a founder of the called the Yuan theory of everything. I believe that's the theory supposed to express any fundamental physical principles in the world. So from my point of view, can you tell me what's the fundamental principle in biology? What is in your field? So I try to use that one, try to see if you can bring me higher from basic physical principles, and I know fundamental chemistry principles, but now to biology is one step higher. So what is the fundamental principle for biology? And you can teach me that one, and then we can gradually let me see if I can find out if you have any questions related. I can use that principle to educate myself.
Yeah, well, you've asked a very difficult and controversial question. The fundamental principle of biology, I
Right.
you will not get any agreement on this among the scientists. I think we are still looking for it. I will tell you what I think, but it's a somewhat of a controversial opinion, and I'm not sure that how many biologists will actually agree with it. But I've not seen a better version. I mean, some people will say that it's something like natural selection through differential reproduction, so evolution. Basically, some people will say that's a basic principle. I don't think that's the basic principle. So what I think is the most useful basic principle is basically cognition all the way down. In other words, I think that cognition is not something that arises late in evolution when brains show up. I think cognition is a superset of life. I think cognition came first. And I think that biology, things we call life, are just things that are very good at scaling up the size of the goals of their parts. So when you have very simple things like particles or molecular networks that operate inside of cells, you have molecular pathways and molecules interacting. Those kinds of things have very tiny goals. Basically, free energy reduction and active inference and things like that. They have very tiny little goals. But biology is what we call systems where there's a multi-scale architecture where they form larger and larger systems where each system has the capacity to work towards bigger goals. So eventually, I call the radius of these goals, I call it the cognitive light cone, which is just the size of the biggest goal that you can pursue. So molecules and cells pursue very tiny, tiny goal states. But the bigger you get, you start to see systems with bigger and bigger goals, potential goals, until you get to humans, which can have like enormous, planetary scale goals, both in time and space, and various other animals have smaller ones. So it's that ability to join into subunits that scale the cognitive light cone. I think that is the fundamental nature of life.
So you said that this biology. So in this case, it's driven. The growth of the biology is driven by the goals. And what the goal? What kind of goal is for the cell and for biology beings? So what are these goals?
Yeah.
Where do they come from? Yeah.
Well, those are two separate questions. What they are and where they come from are two separate questions. So what are they? I can give you some examples. For example, for a single cell, like a unicellular organism, the goals are going to be things like a specific pH range, a specific voltage state of the membrane, a specific metabolic state, a specific shape. Meaning tension around a certain degree of tension around the membrane. They're going to be these very small things, basically at the level of a single cell, that are the goals that it needs to meet in order to survive. When you have a group of cells, they might have very large goals. For example, in a salamander, there's a group of cells whose goal it is to make a limb with 5 fingers. No individual cell knows what a finger is. No individual cell knows how many fingers you're supposed to have, but the collective absolutely does, because if you amputate that limb, the collective will very quickly regrow, right? And within a few weeks, you will get a perfect limb with the right number of fingers, and then it stops. This is how you know it has a goal, because if you deviated from that goal, right? So this is a very cybernetic definition of goals. You deviated from the set point, and it works really hard to get back, and then it stops when it reaches it.
Yes, I have let's see if my interpretation of that goal. So that goal, so if you have a symmetric, right, limbs, if one limb is off, is that possible? The goal for the other limb just copy the other the healthy limb, and because you want to maintain the symmetry, is that possible?
Interesting point. So on the one hand, I would say no, because you can amputate both limbs and they still regrow fine. However, it is true that, and we discovered this a few years ago. If I have a froglet with 2 legs like this, and I amputate one leg, there's a particular bioelectrical state that exists at the wound. Okay, within 30 seconds, the other leg, which we never touch, at the same location, shows the same bioelectrical state. Now, I don't know why. In other words, I'm not sure it's necessary. I'm not sure if they're communicating or not, but it's clear that some information here is not local, that the wound information of one leg was known by the other leg. Does the regeneration use the information from the other leg? I'm not sure. It doesn't seem like it, because like I said, you can cut off both and they both regenerate. So I'm not sure it's that simple, but I just want to say it is true that they pass information, that way. So maybe there's something else going on like that.
So basically, you mentioned about the 2 type. So whether symmetry, are okay. Another is genetic, like a DNA, right? Different embryos. So different embryos grow up. It just exactly looks like a mom, right? All the information encoded in that embryos. Am I right?
Yeah, yes and no. This is a tricky thing. So on the one hand, certainly there are features and disease states that are what they call passed genetically, meaning that your children look like you more than they look like random people, and you can have certain diseases that are passed in families. This is absolutely true. So there is important information passed in the genome, but it is also important to realize that for example, when we make anthrobots, so anthrobots are these amazing little creatures that run around and do various interesting things. They are made from normal human cells with the completely normal human genome. So if I give you the genomic information and it says Homo sapiens 100%, you can't tell me if I got that from a human body or from an anthrobot or from something else. the current way that I now and some other people are now starting to think about this is that the information in the genome is a kind of generative model. It's a prompt. It's a set of cues that are given to the physiology of the cellular collective, and the cells will do their best to interpret parts of that information, to use it how they want. It's a creative problem-solving process. It is not how we used to think is that the genetics determines what happens, right? That it's a kind of driver for what happens. I think much more likely is that it's a set of prompts and a set of resources that the physiology of the cellular collective is using, because we see under novel circumstances the exact same genome can do many different things. And it's not under standard circumstances, it will give you, dogs will have puppies and cats will have kittens, yes, under standard circumstances. But under novel circumstances, that same genetic information will be used in new and creative different ways to make something completely different. So it's kind of tricky. It definitely has useful information, but it is not a determinant of what happens. And you cannot, by looking at the genome doesn't directly specify anatomical features. You can't look at the genome and see anything that says 2 eyes, or that you're going to have eyes, or what symmetry, that it's a bilateral symmetry or threefold symmetry. You can't see any of that in the genome. What you see in the genome are protein sequences, right? The structures of the hardware, the tiny hardware that every cell gets to have. But then it's the physiological software that runs on that hardware that makes the decisions. And under normal circumstances, you see the same thing, but in new scenarios, you will see something quite different.
So you said there's a software. Oh, okay. So in simple terms, so the goal of a cell, is that the goal of a cell is try to survive, is try to duplicate itself, like split the two? Is that a goal for single cell, or you have something more than that goal?
No, I think I think there's more. I think there's more to it, because many cells will preferentially die instead of they will kill themselves instead of dividing in half. We don't know all of the goals of the cell, and the critical point that I make is that you can't just know these by thinking about it. You have to do experiments. So the way you detect goals is by deviating cells from what they're doing and seeing how hard they work to get back to what they wanted to do. So we know some of these homeostatic loops. I'm sure we don't know all of them. For example, I think that it really is one of the goals of cells to be part of a larger collective. Cells love to be part of a group. They love to join. Until things get really bad, if there's poisoning or serious stress or something, they can leave. And this is part of what happens in cancer, is that cells actually disconnect from the collective mind. They disconnect, and then they become like unicellular organisms. So I think some degree of collectivity is also a goal of cells, and then there are many others that we don't know yet.
Are they told by the collective to disconnect, or they do that as a local decision?
Good question. For sure, they can do it as a local decision, and the way it works is there's a kind of a feedback loop. So imagine when you are under normal circumstances in the body, you're a cell, you're part of this electrical network. It's very hard for a cell to do any kind of a calculus about to make decisions. Am I going to stay? Am I going to go? You can't really do that, because as you're connected to everybody else, you have this mind meld. So you cannot really have thoughts as an independent cell, because all the others are sharing their computations with you. So basically, all the computations are around: are we the right shape? Are we the right size? Have we made the right organ? These large scale things. But now imagine that, for example, an oncogene comes in. And one of the first things that oncogenes do is they cause cells to start to disconnect from their neighbors. As soon as the cell disconnects a little bit, it becomes easier to have new computations, because it's not sharing everything with its neighbors, right? It's not feeling the full influx of the signals from the others. So then you can say, you know what? The cell decides, well, I'm gonna try to disconnect even further. So it disconnects more. And that allows it to have even more independent thoughts, which means it disconnects more. And so there's this kind of positive feedback loop that once you start disconnecting from that memory sharing syncytium, then you can make other decisions. And I think that typically happens when when there's a lot of local tissue stress. So cancer, there's a number of ways to have cancer. Mutations are only one of them. And you can imagine that if there is a serious local tissue level stress, cells have an ancient mechanism to disconnect from cells that are poisoned, right? They're called bystander effects. As a cell, you don't want to get poisoned by your neighbor by being too closely connected. If your neighbor has a problem, you want to disconnect. So when tissues are under a lot of stress, that can cause cells to electrically decouple. And the more they decouple, the easier it is for them to make new decisions, such as I'm getting out of here, I'm going, and then they leave, and this is metastasis. Because what happens is the scale of the goals shrinks when you're part of the collective. Your goals are huge. It's like you're making a whole limb. Your goals can be centimeters in size. Those are very large goals. When you disconnect, that cognitive light cone shrinks. That border between self and world shrinks, and now you have little tiny goals about my metabolic state, my energy state, my pH. Am I going to divide, as you said? And then then the rest of the body becomes like external environment to them, right? As the boundary between where do I end and the outside world begins shrinks, and now, as far as they're concerned, the rest of the body is just the outside world, and they're just an ancient unicellular organism in the outside world. So these cancer cells, it's interesting. They're not more selfish. They just have smaller selves, right? Their self has actually shrunk. It used to be this large thing, a whole organ or a whole tissue. Now it's shrunk. So cells can certainly start that process of disconnection. Whether the rest of the tissue can kick a cell out, so to speak, I suspect that's possible. I can't think of a great example right now, but I'm sure it must be possible for the rest of the tissue to also close the electrical connections and disconnect from a cell that's behaving badly. I know one thing that for sure that they try to do is the rest of the cells as a cancer suppression mechanism. The rest of the cells try to normalize each other. So in other words, so we had this set of experiments years ago where we took specific ion channels and we injected them into areas of the body to form an electrical state that says build an eye. Okay, so there's a voltage pattern that says build an eye. And so when you do that, you get an eye. And we did this in the tadpole model, and so you can put it on the tail. You can put an eye, anywhere you want. But what happens then is, so you have these cells that have a voltage that says we should build an eye. And what they do is try to recruit their neighbors to help them build this eye, because we only target a few cells, but you need lots of cells to be an eye. So they recruit their neighbors, like ants and termites. They will recruit their friends to help them solve tasks. So these cells, when they decide that when we push them towards making an eye, they recruit their neighbors. But at the same time, their neighbors are trying to push them not to be an eye, because as far as their neighbors are concerned, no, you should be normal skin or muscle or whatever the region is. And that's a cancer suppression mechanism. If you have a cell that has a weird voltage, the neighbors are going to try to normalize that voltage through these electrical synapses. So so what happens is there's like this battle, and it's not a battle of the cells, it's a battle of patterns. It's a battle of goals. You have one pattern that says we should be an eye. There's another pattern that says no, you should be skin. And basically it's the patterns that compete, and eventually one will win, and either you'll have the ectopic eye or you won't.
And those patterns are expressed by voltage.
Those patterns are visible to us as spatial distributions of voltage, and we have the ability to see them directly. So we can see them, and we can rewrite them. At least in many cases, we can rewrite them and make other things. We can make extra hearts and brains and limbs and some other stuff.
But they're not. That doesn't mean they are the voltage. That's the expression that we have that we can interpret.
Well, that's a subtle philosophical question. What we have access to, what we observe, is the pattern of the voltage, and it's the voltage pattern that determines what happens. Yeah. So by changing the voltage, you can control what happens. But I think, as we said before, I think what's ultimately driving this are certain facts of mathematics that exist outside of the physics of this world that actually determine what kinds of patterns are going to be formed in cells that have specific types of ion channels, right? If you actually ask the question, well, why is it this pattern, not that pattern? The answer eventually boils down to math. You'll just eventually come to, much like with Turing patterns and chemical signals in embryos, eventually you just get down to the answer is math. Is that ends up being the answer.
One of the reasons why I reached out to you is, Doctor, you showed me, I guess it's in some other countries and maybe even here in America, they're doing stuff with wearing garments and things that can help with electrical voltage for cancer. Have you heard of this? I'm sure you're familiar. This sounds like right up what you're talking about.
I don't know about any specific products. I will say that uh, anything's possible, but external application of external electric fields or things like different materials to modify electric fields, all of that. Maybe that has effects, but that's not the system that I've just been describing. The
I
system
understand. Yeah. I'm just,
that I've been describing is very difficult to affect with external kinds of things like that, and so I don't know if there are such devices, maybe, but I don't know anything about them.
Uh, yes, so there's some kind of standard therapy. They wear a jacket, back and the front, so they have they apply the little voltage difference. And whenever you have a voltage difference, right, and it creates some kind of basic vibration, motion. And since you said apply different voltage will be changing the goal of the cell, which I think that's very logical, because of the voltage, that's the power, that's the energy drive cell grow or drive cell split, develop. So that's the energy required. However,
but just to be clear, sorry, just to be clear, it's not just an energy thing, because it's the actual spatial pattern, right? It's the same,
of course, yes.
Right? The information is carried so of course you need energy for sure. If you don't have energy, the whole thing will die, right? You need energy to make the ion pumps go. But it's the there are multiple patterns that require the exact same amount of energy, but these patterns have very different meaning to the other cells, and they make different organs. So
exactly,
right. So it's not a submissive energy thing; it's an instructive patterning thing.
Right. First of all, you said that you have to apply voltage to
we don't apply any voltage, right? So something else. We never apply any voltage. So what we're external, I mean, people there's a very long history of people using applied voltage to make certain kinds of effects. This is not what we're doing.
Oh, okay.
The cells naturally have these ion channels that can be opened or closed in different combinations. And this gives rise, according to some laws of mathematics, this gives rise to a specific pattern. It's an excitable medium, and it gives rise to specific patterns. What we do is we open and close the channels. In other words, there's an electrical interface that the cells are already using to talk to each other. And so what we either use optogenetics, or we use pharmacology, we use different ways to basically control, like every cell basically on its surface, it has a bunch of control knobs that we can tune. And that will, and we need and it's quite complex. You need a computational simulator to know what to open and what to close, which we have developed. So there's a computational simulator that tells you that if you want a pattern like this, then you're going to need to open and close certain channels. So
how do you open and close the channels?
There's a number of ways. The easiest way is by pharmacology. So there are drugs that will open and close specific kinds of channels. There are many drugs like that. Also, there's something called optogenetics, because some channels can be turned on and off with light. And so this is what the neuroscientists use when they, control neuronal firing with laser light. So we've done that too. We've done that in cancer. We've done it with birth defects. You can turn specific channels on and off with light.
Okay, so you can turn on with the light. The difference between light and electricity is only the oscillation frequency difference, and then also the energy level are different. So can you turn on and off with electricity?
You can't turn on and off with applied electricity. You can't turn on and off individual channels. What you can do is provide long range electric fields. So if I put 2 electrodes on my system, I will have an electric field. And many kinds of cells are very sensitive to these electric fields, and so they will crawl. So, for example, directionally, right? So some cells love the positive, some cells love the negative pole, and so if you want to, for example, neurons are like this. If you want to get your neurons, and there's some very beautiful people like Min Zhao and Colin McCaig and other folks over the years have shown these incredibly beautiful movies where you have cells wandering around in a dish, and you turn on the current, and boop, they all start going. Then you flip the polarity, then they all start going this way. So in the body, this is used when you want to direct cells to a particular region. There are natural electric fields that will direct cells, but it's not the same. It's a different set of phenomena than to set up a pattern that makes for an organ or a bigger structure like that.
Yeah, that's one of the principle of the electrical voltage therapy. So if you turn, of adjustable to the human level, accessible level of voltage, you would align those cells, right? Align those cells, because every cell have a voltage, which means it has north and south, positive and negative polarities. So when you apply external field, electromagnetic field, so it will align cell. So it will, I believe it will have some kind of health benefit. Make all the cells towards common direction, common shape, instead of like cancer grow wildly.
Yeah, I mean, so first of all, there are some cells that have different polarity on one side on the other, and they can be aligned. There are some cells like that, but the vast majority of cells have the same voltage all the way around. In other words, the voltage is between inside and outside, right? So if you have a cell, the voltage you measure is like this across.
Of course, that's great.
So it's not here; it's here, right?
Exactly. You have to cross the cell
exactly
membrane to have a voltage. Yes.
Exactly. So most cells have a similar voltage all the way around
on the surface, right?
Some cells have different. One end is more positive than the other end. And the thing about these applied kinds of fields is maybe there are applications that have health benefits, maybe, but typically when you do these kinds of things to a complex system, it's much easier to break things than to fix things, right? It's always easier to break things. So you have to be really careful, because in a system that's using these kinds of signals to orchestrate complex cooperative behavior, it's very simple to introduce signals that are going to confuse cells and so on. But yeah, I mean, I'm sure it's possible to also have beneficial applications. It's the trouble is that applied fields they're kind of a blunt tool. They provide one bit of information. That way, they align. They can, but what the cells really want are complex patterns that are going to say which organ, how big, how many fingers, how many eyes, right? And that's very hard to do with electrodes. Maybe someday somebody will figure out how to do it.
How do you use light to activate it? You have to do it in laboratory, but inside of a human body, the light cannot go through it. Is that right?
No, yeah. So that's right. So currently, well, there's actually two problems. One problem, and remember that the things I'm telling you, we don't do them in humans yet. So this is all in animal model systems in the laboratory where we have the ability to do these experiments. There are actually two problems. The first problem is that the light sensitive channels are not native to most species. In other words, we have light sensitive channels in our retinas, but the rest of the body typically doesn't have that many. I mean, there are some exceptions, but typically we don't. So that means we have to introduce light. If we want to do these experiments to prove that the bioelectrics is doing something, we have to introduce light sensitive channels into the animal. Right. And so we get these channels, we get them either from retinas or from other creatures. There are some creatures that have these rhodopsins and things, bacteria often have them. So we can introduce them. So that's problem number one, is that you have to have them. So normal humans walking around, I think the vast majority of your body does not have optogenetic sensors in it. The other problem, as you point out, is that the light doesn't penetrate very far. Now, some of these things are activated by far red, so you get a little bit of penetration through, but not really. And so I think the actual applications in human medicine is going to be more pharmacology and not so much optogenetics, I suspect.
Well, certainly, that's where the money is.
I mean, well, yes and no.
I'm just saying.
Yeah, just to be clear, the vast majority of so something like 20% of all drugs are ion channel drugs. And the vast majority of them that are being used for other purposes, neurological disease, cardiac disease, gut, inner ear, things like that. Many of those things are off patent. They're extremely cheap drugs. So a lot of these ion channel drugs are very cheap. And so as a therapeutic, this is not the same as some kind of $1 million stem cell therapy. These ultimately, when these drugs are deployed in humans, it's going to be a very cheap therapy.
Are you familiar with Dr. Thomas Seyfried at Boston College, his work? And
Yeah.
Michael Lasanti, his work at Southwood University? He's
Nope.
using doxycycline and azithromycin for cancer, breast cancer patients. And
No, I don't know. I mean, there are many, there's
yeah, I know. These are just folks that we've interviewed, but I'm just curious if you're familiar.
Yeah. Thomas Seyfried is great. I love his work. I think he's really good.
Yeah. Is there an understanding, kind of like the bioelectrical level, of what's going on with his proposed mechanism of treating the cancers with?
Well, his is metabolic, right?
Right, but there's always an electrical component to that, wouldn't there be?
Probably, I don't know. We have not unified our approaches yet. So yeah, but it's clear. It's very clear that he's onto something. That much is clear to me.
Yeah. Well, I really appreciate your time. I thought
you.
it was a great opportunity to kind of introduce some of these ideas to our audience and to learn more about what you're doing and how these kind of correspond with some of the things that we've been learning along the way in our program. One of the things that I don't know if you know if you know of the late biologist Ray Peat. Have you heard of him? He talked often about these anecdotal stories, where little children have their finger severed, the whole tip of it, and then if you don't expose it to oxygen, put like a little cigar cap, all these stories of them regrowing.
Well, this is true. Children regenerate their fingertips. Yeah, that's known. That's true.
Why is that? Why is that? And how come, it's so that effect that salamanders express so easily is so hard to express for humans? What's going on there?
It's a good question. Now I'll point out that some mammals, for example, deer. Okay, so deer every year they shed their antlers and then they regenerate. Deer, when they're regenerating, they regrow up to 1.5 centimeters per day of new bone. Can you imagine that? 1.5 centimeters of new bone per day. It's incredible. So here's a large adult mammal that can regrow bone, vasculature, innervation, skin, all of the things that it takes to make an antler rack. They're regrowing. So it's not as if mammals can't do it, but humans typically are not good regenerators other than liver. Yeah. So we regenerate our livers. Otherwise we're not very good. It's not entirely clear why mammals are so much worse than generally, except for deer, are so much worse. But I can give you a story that may be true or may not be true. We don't know. Imagine that you're an early mammalian ancestor. You're like similar to a mouse. You're running around the forest, and somebody bites your leg off. Well, the problem is, are you going to have time to regenerate anything? You're gonna A, bleed out, B, you're going to get it infected, C, you're going to be stepping on it, which means grinding it into the dirty forest floor, right? Nothing's going to regenerate under those conditions. I think it's interesting that the one example of a mammalian regeneration is one you don't put any weight on. It's the antlers. Yeah, there's no, you can keep that just sort of exposed and nice and clean, and you're not trying to put weight on it. So I suspect that evolution, basically at that point, because of our new lifestyle, evolution went the scarring route. Seal the wound, scar, inflammation, kill the bacteria, and hope you survive. That I think is where evolution went with this, as opposed to a salamander, which has a nice aqueous environment. They're buoyant. They don't have to put any weight on anything. They can sit there with their low metabolic rate and kind of take 3 weeks to regenerate. They don't need to be running around and have this like high metabolism. So that's a story that I think makes sense, but nobody knows if that's actually why it got shut off. But I don't think there's any, it's very clear that mammalian embryos, including human embryos, know how to build a human. They did it once during embryonic development. Those processes are still there. And so if we knew how to activate them, I think we could get basically some very transformative regenerative medicine. That's our hope.
Wow. That'd be fantastic. When you see, I wanted to ask you briefly, is there any implications for your ideas in regards to, I'm a fan of Rene Girard's mimetic theory and the idea of human mimesis and the patterns of why humans tend to, and not just humans, animals too, to some degree, mimetic desire, where they desire what their neighbor they perceive to be desiring. Do you see any of that with some of the different topics that we've explored today? I'm thinking about the Platonic space, or there's there seems to be an interconnectedness to human beings. And Rene was, he was an anthropologist from Stanford, so he didn't dive into the biology. But I'm always looking to talk to biologists to see if they see anything that kind of links up with the patterns of why we're so mimetic and why we sync up like that.
Yeah, I don't know the work, so I can't say anything specific about it. What I do think is that all of us, and it's not just humans, but it's basically all active agents, not even just biologicals, but all of us are fundamentally linked by our dependence on these fundamental truths of mathematics, computation, and other things. We all have access to the same to the same patterns, basically. And so I think we share that vertical descent from these laws of these laws of mathematics that are kind of like the vertical descent of the evolutionary tree. They're kind of like that. We all have that root in common.
But Dr. Yu, any final thoughts before we close out?
I do have that one before the end for Professor. There is one way, one called cancer therapy. They try to activate the killer T cell. T cell, they call the killer cell, killer cell. To activate the killer cell, that's somehow your natural body's immunization cell, and help you to kill any foreign objects like a cancer somehow. So, is there any biological method, to activate the T cell and suppress some kind of cancers growing cell? Is there any method? So that could be some kind of significant in cancer cure.
Yeah, well, there's a very large field of cancer immunology which works on exactly this. So the idea is to get your immune system to kill the cancer cells. There are many people working on this. This is a large area of oncology. The problem, of course, is that it's not foreign material. These cells are your own cells. They have all your own materials. They have all your own epitopes.
Right.
So distinguishing them is really hard. This is the challenge of the field for the immune cell is to educate the immune cells on what it is that they should be killing. Because if you get it wrong, they're going to kill a bunch of your own good cells and then precipitate a crisis. So, but there are many experts on this field. Cancer immunology is a very nice growing field. So I'm quite optimistic. I think they'll have some good successes, I think.
Well, very good. I appreciate your time.
Thank you so much.
Thank you.
Yeah, it's great to meet you both. Thanks very much.
Right. Thank you.
Be well.
Thank you for teaching. Okay. Sharing your knowledge.
Thank you for the conversation. Thank you. Bye.